So we'll start understanding these steps. This is called as the viral multiplication. Also called, you can call it as a viral reproduction. Viral reproduction and also called as the viral multiplication. Why we are not calling the reproduction strictly? The reason is that this is not a reproduction, actual reproduction. Fine? Fine. So this is the viral replication. Fine. Start with the adsorption. So what is happening? Suppose there is a person, where I should write here, there is a person, we can take, it is your classmate Shivam, okay, he is having the 2 eyes, 1 nose, 1 mouth, yes, 2 legs and 2 feet and 2 hands, fine. Now, he is having the common cold. Suppose he is having the common cold. Don't talk. If you want to talk, talk with me. It's a common cold. So he is having a common cold, and the thing is, he is having one friend. Any best friend of you in classroom? Razan. So he is having the another friend called as the Razan. Okay. Initially, he won't have any common cold, but since they both are the best friends, so they are living in close, okay, and the what is called as the Shivam and Razan. Shivam is having the common cold, so he sneeze, and you know the sneezing, what is it? The droplets comes. Yes, air droplets comes. So this air droplets, it is because he is enclosed, so it is inhaled by the Razan. So now we will see that he has already suffered. He has already suffered from the common cold. Now this, you can say, what is this? Droplet. This droplet is going inside the Razan nose, he inhaled. So what actually happening with the Razan? Now we will see the story. What happening with the Razan? So Razan inhaled. It's inhaled, the particles goes inside his respiratory tract, okay, and you know that respiratory tract is lined by the epithelium, respiratory epithelium. So we will take one epithelium. You know that epithelium is what? It is a tissue. It is a tissue, and tissue is made up of from the cells. Yeah. So we'll take one epithelial cell, not tissue, cell. Fine. Yes. So we'll take the cell. Yeah, so this is suppose a epithelial cell. I'm not making the exact diagram, just a rough functional diagram. So this is the epithelial cells, and you know that the cells is having the brain, and that is called as the nucleus. That is called as the nucleus. You know that in the cell membrane, we are the eukaryotic organisms. We are the eukaryotic organisms. We won't have the cell wall. We won't have the, what we have is the cell membrane. What we have is the lipid bilayer phospholipid. Basically the membrane is made up from the phospholipid, and the thing is, our cell won't allow anything to come inside and anything goes out. Yes, our cell is highly selective and semi-permeable. Highly selective and semi-permeable. It won't allow anyone to come, any stranger to come inside basically the house and make the disturbance. They won't allow. Okay. So basically our cells keeps the gatekeeper, a security guards, gatekeeper or security guards. So this epithelial cell is also have the gatekeeper. Okay, different gatekeepers. Fine. Now, one of the gatekeeper is meant for this viral droplets. Okay, viral droplet is basically having some viruses. It is inhaled. Now it goes inside his respiratory tract. We take in the one cell of the epithelium, we put it here, and we seen that our cell is not you can say not welcoming everything to comes inside. Okay, it's have the gates, it's have the gatekeeper where there is a security guard. So one of the gatekeeper, it is meant for this viral droplet. Okay, viral droplet. Now this viral droplets becomes virus by the filtration. It's become the virus, and it's come here and it starts sitting over his place. Okay. This process is called as the adsorption. Since it is happening on the surface, we are not calling as the absorption, we are calling as the adsorption. There's a difference between the adsorption and absorption. So this is the adsorption. Clear? Now, as soon as this what is called as the drop virus comes over the receptor, this is gatekeeper is called as the receptor. So it comes on the receptor, it triggers the intracellular signaling. कि आप उसके ऊपर में आ गया है, वो आप अंदर का चीजों को बोलेगा कि हमारे ऊपर कोई आके बैठ गया है, तुम लोग रेडी हो जाओ, मे बी ये हमारे लिए यूजफुल होगा और हमारे लिए यूजफुल नहीं होगा. द थिंग इज our internal structure of the cell, okay, it's phagocytose. This virus, phagocytose means engulf, okay. Now, okay, so this comes inside, okay. Now viruses comes inside, of course, this is the nucleus and there are ribosomes and other organelles too, fine. So our cells, what we did, अब इसे अपने अंदर में इसे ले लिया है. This process is called as the penetration. This is called as the penetration, and the process by which our cell engulf the virus, it is called as the endocytosis. Since it is happening inside, it is called as the endocytosis. It is a part of phagocytosis. Have you heard about the phagocytosis? Phagocytosis is basically performed by our cell as well as the cell organelles like the macrophages. Basically whatever external thing is coming, okay, they start to engulf them. उसे वो खा जाता है, okay. So engulfing inside is called as the penetrations. Now what is happening? The next step. This our cell will close the virus, okay. Now here is the nucleus. Here is the nucleus, and now viruses, because you know that our cell is having the lysosome. Yes, our cell is having the lysosome, and lysosome contain hydrolytic enzymes. बहुत powerful enzyme होता है. It's like an acid. अगर आपके हाथ पे एक बूंद गिर जाए तो आपके हाथ को जला देगा. That much strong enzyme is carrying by your lysozyme. Okay, it is the acidic enzymes called as the hydrolytic enzymes, and there are different enzymes. What lysosome is doing, वो क्या करता है? तो you know that viruses is having what? The capsid. Isn't it? We discussed, no, or some viruses do have, along with the capsid, they have the envelope. तो उसको तो हटाना पड़ेगा, because the main infectious things is what? Their genetic material, the genome. So क्या करता है lysosome? तो lysosome वायरस के कोटिंग्स को, जितने भी कोटिंग था उसको क्या करता है? हटाना स्टार्ट कर देता है. Now the coating is start disrupting. Yes. And viral genomes are now free. This step is called as the uncoating. Lysosome ने इसको क्या किया? हटा दिया. Now we move further ahead, and what is happening? अब ये जैसे ही, as soon as this become free, the viral genome become free, it start instructing our nucleus कि ब्रो अब तुम मेरे लिए काम करना स्टार्ट करो, ओके, तुम मेरे लिए काम करना स्टार्ट करो. तो ये इजी नहीं होता है, बिकॉज तुमको मैं बोलूँगा तुम मेरे लिए जाओ 2 लीटर पानी लेके आओ, 2 किलोमीटर दूर से तुम मेरे लिए 2 लीटर पानी लेके आओ. देन अगर बेसिकली तुम अपने मर्जी का मालिक हो, मैं अपने मर्जी का मालिक हूँ. इन रियलिटी यस, तुम अपने मर्जी का मालिक हो. ऑल्दो नाउ आई एम योर टीचर, सो यू कैन लाइक तुम मेरे बात मान लोगे. बट सपोज इफ यू मीट विद द स्ट्रेंजर एंड दे आर कॉलिंग यू कि तुम मेरे लिए ये काम करो, then you will not suddenly be ready. No, you suddenly not be suddenly ready. You will take time. And you will see that okay, if it is beneficial for me, I am ready. If not beneficial for me, I am not ready. लेकिन ये जो वायरल जीनोम होता है, ये क्यों कन्विंस कर लेता है? Because it is having the some, you can say, the enzymes or some genetic यू कैन से द मैसेजेस जो इसका क्या करता है हमारे न्यूक्लियस को हाईजेक करने में मदद करता है. आई एम नॉट गोइंग इन डिटेल बिकॉज देयर इज अ सेपरेट ब्रांच कॉल्ड एज द वायरल जीनोमिक्स. ओके, उसके अंदर में तुम डिटेल में बैठ के यही चीज सब पढ़ोगे. Are you getting my point? But for your exam point of view, through the viral genome, our nucleus is hijacked. Now it becomes the viral factory. It's become the viral factory. So अब अगर ये viral factory हो जाएगा, then it will start replicating. So this is our nucleus. Okay, and our nucleus do have the genetic material, and it start helping the viral genome, और ये अपना नंबर बढ़ाता हुआ जाएगा. Fine. Now at one time comes, they all will assemble, okay, and it start releasing. It start releasing, coming out from the one cell. अब एक सेल को तो इन्फेक्ट कर दिया, no? What is the his viral tendency? क्या वो दूसरे सेल को इन्फेक्ट करेगा, isn't it? तो अब क्या करेगा? तो यहाँ से ये रिलीज होगा. It will release from this, release by either the budding. Okay, either the budding or either by the rupture. So the thing is कि अगर if it comes out through the budding, okay, outgrowth, then वो क्या करेगा? तो इस मेम्ब्रेन को चुरा लेगा. Are you getting my point? It will steal this membrane. It's like a thief. Okay, viruses is like a thief. If it will comes by this mechanism called as the budding, outgrowth, it will thief this membrane. It will steal this membrane. Okay, and once it will steal, it's become what? It's become the enveloped virus. It's become the enveloped virus. So that is the concept behind having the envelope and not. Okay, so envelope is not a viral, you can say, on cell formation. No, it is basically still ये चोरी किया हुआ membrane है हमारे ही cell का. Are you getting my point? Yes. Now, if it is coming through the normal release, it won't have the envelope. So it is the non-enveloped virus. Clear? Yes. Understood? So this is all your mechanism. This is called as the assembly. This is called as the release. This is the viral multiplication. Getting the point? Yes. So viruses do not put their own effort to multiply or increase their number. They are dependent upon the host cellular machinery. Clear? Yes. No problem. Fine. So let's conclude today's class. This is all about your virus. Your virus is finished. Okay. And the few things you just remember. This is the first limitation of the Koch postulate. Okay. We discussed Koch postulate. So this is the one limitation. Second thing is it won't have any place in the five kingdom classification given by the RH Whittaker. Okay. It won't have any accurate place. So this is the exception of the RH Whittaker 5 kingdom classification, as well as it is the exception of the Robert Koch postulate. Clear? Yes? Fine. So concluding today's class, what we studied about the introduction of the virology. We discussed about the viral's morphology, that is the shape, size and their structure. We discussed about the classification of virus, and at the last we discussed about the viral replication, also called as the viral reproduction. So this is all about your today's lecture. Tomorrow we will start with the fungi. Okay, fungi, and again one further class for the protozoa. Then this chapter will finish, and then we will move ahead and discuss about the immunity, the immunology. That is your last chapter. Okay, so till 14 I will take your class, then your class will finish. Now I am giving you the 5-minute time, 5-minute or 3-minute time or 2 minutes, whatever you take. You write the feedback for me. Okay, whatever you finding the merit or advantage or good thing about me, and what you want. Listen. What you want correction in me, like तुम्हें क्या मेरे में अच्छा लगता है पढ़ाने, कम समय क्या अच्छा नहीं लगता है, क्या तुम चाहते हो कि मैं ऐसा करूँ जो you will learn the better way. Fine, write it down. Take your paper, a small paper, write it down. If you are comfortable, write your name. Otherwise, no need to write your name also. Clear? Start writing. So thank you for having me. Bye. Have a nice day.